In Chapter 4, we're going to discuss the genetic code, mutations that can happen to this genetic code, and also translation, the machinery by which RNA information is translated into a protein sequence inside the cell. So the genetic code. The genetic code is basically a dictionary that tells the cell which codons on the mRNA are going to encode which amino acids in the growing protein. Do you have to remember the genetic code for test day? Absolutely not, okay? I think if you're ever going to get a tattoo, this would be a cool one to get because you just never know when you might need the genetic code. Which bases encode a particular amino acid? Just kidding. All right, now let's take a peek at what this genetic code can tell us, and let's make sure that we know how to read a codon table in case we need to do it on test day. As I had said, they would give you the table if you're expected to figure out amino acid sequence. So, what you'll notice is that the first position of a codon is going to be this box over here on the left. The base in the second position of the codon, you're going to have to choose one of these boxes in the middle. And then for the third position of the codon, you're going to need to choose one of these boxes on the right. So, for example, U. We would choose U over here because it's in the first position. So right now we're narrowed down to these 16 choices. The second position is U. So now we're narrowed down to this box. And we're narrowed down to 4 choices. And then the third position being U. Now we know that this is going to correspond to this top box position. So U. And we can safely say that this would encode phenylalanine. All right, let's practice another one. What is UUA? How would we go about doing this? Let's look at the table. The first position. Now you could search through, if they give you all the bases, you could search through and look for UUA, but sometimes they don't put the bases in like this. They would just give you the positions that these bases should be in, and then you have to figure out which amino acid would be there. So what is UUA? The first position is U. So we're narrowed down to this top box of 16. The second position is U. So we're narrowed down to this box of 4. And then the third position is A. So we're narrowed down to right here. And we can say that UUA is going to encode the amino acid leucine. Something you should notice right now, and that is that when you look at these bases, the first 2 bases and the third base are a little bit different in terms of their specificity. If you see the first 2 bases, oftentimes are all that's required to encode an amino acid. Look at this down here, leucine. Cu. It doesn't matter what the third base is. It could be a U, a C, an A, or a G. They're all going to encode leucine. So the take-home message here is that the third base provides much less specificity for coding an amino acid. This is called the wobble hypothesis. So what this means for the test is when you're asked about mutations that happen to sequences of RNA or sequences of DNA that encode RNA, if the mutation's to the third position in the codon, that's going to be less likely to cause a detrimental mutation which changes protein sequence. We'll review that fact again shortly. Let's practice one more. What is CAG? How are we going to figure it out? Let's go here. We see that the first position is C, so we're limited to these 16. The second position is A, so we're going to come over to here. We're limited to these 4. And then the third position is G. CAG, you see, encodes GLN. Is that glutamate or is that glutamine? Ah, it's glutamine. Remember, GLU is glutamate, GLN is glutamine. You can remember the GLN because the amino group, the amide linkage that's present in glutamine. By the same token, ASP is aspartate, ASN is asparagine. Okay, so just keep that in mind. So yes, CAG is going to encode glutamine. Having mentioned CAG, what does CAG mean to you? It should mean something to you. at least by the time you're done preparing for the examination, you should remember that CAG repeats are seen in what disease? Huntington's disease, right? CAG trinucleotide repeats get expanded, and you see those in Huntington's disease. So in the protein Huntington, encoded from the Huntington gene, you see extra glutamine repeats within that particular protein sequence. So some of the properties of the genetic code that you're going to need to be familiar with for the exam are the following. Number one, this code is universal. Now, what does that mean? That means in all organisms that you're going to see, if it's viruses, bacteria, parasites, eukaryotic cells, the genetic code is going to apply to all those cell types. There's one exception where the genetic code doesn't hold very tight. It holds, but there might be some nuances to that, and that's in mitochondria. Now, you don't have to worry about those differences. Just understand that the genetic code, it's universal. You could get that tattoo, and it would apply to any organism that you're going to look at, except for maybe mitochondria. The code is degenerate. What that means is that one particular amino acid can be encoded for by more than one codon triplet. So UUU, UUC, they both encode phenylalanine. So the genetic code can be degenerate. Now, what we mean by unambiguous is that one codon triplet, is always going to specify the same amino acid. That doesn't change. Now, the third base might get mutated and the same amino acid is specified because the genetic code is degenerate. But the same triplet always specifies the same amino acid. When you look at the genetic code, you see that there's 4 possible bases and that there's 3 places for those bases to go. So from that, we can mathematically derive that there's 64 possible codons. 61 of these, it turns out, encode amino acids, and 3 of these don't encode amino acids. 3 of these encode stop codons. These are the ones you have to memorize. So, the ones that you have to remember are 4. 4 to remember. You have to remember the start codon, which is an AUG. Now, in RNA, that's going to read AUG. If you're looking at the coding strand of DNA, that's going to read ATG. So keep that in mind. AUG if you're looking at RNA, but if you're looking at the coding strand of DNA, you're going to be looking for the ATG to correspond with the start codon. The other 3 to remember are UAG, UGA, and UAA. You need to commit these to memory, and there's a reason for this. You'll see shortly. UAG, you can remember you are gone. UGA, you can remember you go away. And UAA, you can remember you are away. Right? Pretty soon biochemistry will be away. It'll be away forever once you get past step 1 of the boards. Now, UAG, you are gone. UGA, you go away. And UAA, you are away. When the ribosome comes along and sees one of those stop codons, it's going to go away. So this will help you remember.